I'm your host, Nick Jikomis, and today I'm speaking with Dr. Leonard Guarente. Dr. Guarente is a biology professor at the Massachusetts Institute of Technology, where he's been running a lab for many years. His scientific work focuses on the molecular and cellular processes involved in aging, including the molecules and the genes and all of the cellular level stuff that's involved in natural aging, as well as ways that we can develop strategies to actually extend the lifespan by tapping into some of these biological mechanisms. Dr. Guarente is also interesting because he's the founder and chief scientist for a company called Elysium Health, which focuses on taking the discoveries that are happening in aging research and actually bringing them to the public, both in the form of creating new health supplements, which can address various aspects of cellular, immune, or cognitive aging, as well as technology products that actually help you learn about your own aging, the rate at which your body is actually aging. And we got into a bunch of different topics related to aging and how all of these different things work. We talked about a molecule called NAD and some of the sort of core biology, the metabolic biology involved in the aging process and how that changes across the lifespan. We talked about molecules like omega-3 fatty acids and resveratrol, the famous molecule associated with red wine, as well as other molecules like pterostilbene, which have enhanced bioavailability properties that might lend themselves to be better used in health supplement products. We talked about Elysium Health, the company that Dr. Guarente co-founded, and how they use the scientific research discoveries from his lab and from other labs to actually engineer products, health supplements, that have ingredients that are most likely to address various aspects of aging, whether that's at the level of cells and metabolism or the immune system or the brain. So we got into all that stuff. We also talked about epigenetics, the idea that you can actually look at certain molecular signatures on the DNA to determine or calculate someone's biological age in comparison to their chronological age. So for example, I took a test that measures this through a product that Elysium Health sells, and I learned that my biological age is actually 30, but my chronological age, the number of years I've been alive, is 34. So my rate of aging is a little bit slower, the rate of biological aging, than my actual number of calendar years would suggest. And so we talked about how some of this technology works and how some of these supplements are utilizing findings from the scientific literature to actually try and build products that people can use to enhance or extend their lifespan. So if you're interested in aging biology and some of the practical aspects of that, things out there that you can do to learn more about your own aging, what's going on in your own body, or supplements you might be able to take to address aging-related decline for yourself, this will be a really interesting episode, and I definitely learned a lot from speaking with Lenny. And as always, if you enjoy the content of this podcast and you're learning a lot from it, please do like, share, and subscribe. I appreciate 5-star reviews on Apple or Spotify or other podcast directories that you may be using. You can subscribe to the video version of the podcast on YouTube or on Odyssey. And you can also sign up for my free weekly newsletter at mindandmatter.substack.com. You'll get a weekly update about the podcast, as well as a bunch of other interesting research and other things that I'm looking at related to the topics I cover on the show. You'll have access to my long-form science writing on Substack. Hey everyone, I want to take a moment to tell you about Double Blind, one of the leading media and education platforms dedicated to psychedelics. They provide news, information, and educational resources about a range of topics related to psychedelics, including policy, science, culture, and more. They partner with leading experts to help produce high-quality content. They have a biannual print edition, which comes twice per year. It's beautifully produced with great photography, artwork, and packed with lots of interesting information. Double Blind is also online with monthly webinars with experts and a variety of affordable online courses about all kinds of topics related to psychedelics. If you go to the link in the episode description, you can get 20% off site-wide. All proceeds go directly to support Double Blind's independent journalism and destigmatization efforts. So visit doubleblindmag.com/mindmatter for more information. Today's show is brought to you in part by Dosist, an all-natural cannabis company specializing in dose-controlled cannabis products made with plant-based ingredients. To learn more about Dosist, their products, and where they are available, please visit their website through the link in the episode description. And with that, here's my conversation with Leonard Guarente. Dr. Leonard Guarente, thank you for joining me.
My pleasure.
Can you start off by just telling everyone a little bit about who you are and what your scientific background is?
I am a professor at MIT. I've been on the faculty there for a long time, 40 years or so. And my background is in molecular biology, including biochemistry and genetics. I have studied a few different areas of research with my lab over the years. Initially, we worked in the area of cell biology, and we specialized in the organism of yeast, which are single cells and relatively easy to study, but which teach us broad principles about biology. And we did that for about 10 years or so. And at that point, I got a little bit antsy to try something a little bit bolder. And we decided to make a foray into research on aging. And this was a very kind of checkered past, riddled area at the time. And the balance was, on the one hand, it was a area of not extremely high repute. And on the other hand, there was an opportunity that if you actually did make significant progress, it was a pretty big deal because there was really not much known about it except descriptive things, comparing old organisms with young organisms and seeing what changes with aging. So that was essentially the approach that had been done in the decades prior to the early 1990s when we got involved in this. And so we worked in this area for some time. And since my lab was very experienced with yeast, we made the decision, and it seemed ridiculous to some people at the time, but we made the decision to begin our studies of aging by studying aging yeast cells. And that's what we did. And pretty much the first 5 or 10 years in studying aging, we were studying aging in yeast cells. And what we set out to try and query was whether there were specific genes, and we expected a small number, which somehow regulated the rate of aging in yeast cells. And we decided to approach that problem by working using an assay for aging in yeast that was not measured by a clock or a calendar. So it was not, you were not measuring time, but you were measuring how many times cells divided. Because it was already known that yeast cells had a very limited number of divisions, roughly 20 or so, after which they stopped dividing. And what we then did was watch yeast cells divide under the microscope and count the number of times they divided. And it was about 20 or so. And we wanted to identify variants, which are otherwise in genetics known as mutants, which would be changes in the DNA sequence in the genome of yeast, which would have the effect of allowing that variant or that mutant to divide more than 20 times. Okay, so that would increase the lifespan, if you will, of that cell. And that really took about 5 to 7 years to get through this, to actually succeed in identifying genes that had this property. And and it was very tedious work in the beginning. But an interesting gene came out that we really have been studying ever since, which is a gene called SIR2 in yeast. And it turns out that this gene is found in pretty much every organism or species out there, and that includes simple animals like worms or fruit flies, and mammals such as mice or humans. So we all have genes that have a similar sequence and encode similar proteins to this yeast gene, which is called SIR2. And in nature, collectively, these proteins have come to be called sirtuins because of their similarity to the yeast gene SIR2.
And what does this gene encode? What is the resulting protein doing?
Yeah, so the gene, that was what we're interested in. So what the genetics told us was that if you eliminated this gene from yeast cells, you made their lifespan shorter by about half. Whereas if you gave yeast cells an extra copy of this gene, so they made more of the protein than normal, they live longer than the natural strain, which in biology is called the wild type strain. So that really said that this protein had the ability to regulate the lifespan in yeast, which is this peculiar lifespan based on cell divisions. So we started the next period trying to figure out exactly what the SIR2 protein does. And we were studying two proteins, one from yeast, which was the SIR2 protein, which we purified, and the closest homologous protein from humans, okay, which is a protein called SIRT1. Okay, so these are both sirtuins. They're similar to one another, but not identical. One comes from yeast, the other comes from humans. And we purified the human protein as well. So we had these two proteins in a test tube. And we had some background information that allowed us to focus on a process which is mediated by sirtuins, which is called silencing in yeast. And silencing is a process which takes a region of a chromosome where genes can be expressed in this region, and silencing shuts that down so that the genes are not expressed. So it's a kind of process that results in a portion of a chromosome just being kind of wrapped up in an inactive state. So it doesn't do anything, okay? And it turns out that silencing is an aspect of something that has since then become pretty prominent in biology called epigenetics. And so genetics, as started by Gregor Mendel in the 1850s, is based on mutations that change the DNA sequence of a gene and change the amino acid sequence of the protein encoded by that gene, okay, to result in a change in something you can see in the organism. Okay, in Mendel's case, it was peas and plants and flowers. In epigenetics, okay, you also have a change that you can see in the organism, but it is not mediated by a change in the DNA sequence at all. It is something, it's different from that, which is where the epi comes from. And what it turns out is that it's a process that takes the chromosome and just shuts it down, okay? And it was known that there are proteins that bind to the chromosome that are called histones. And histones are essentially just wrap the DNA into a ordered structure across the genome, such that the histones form a ball of proteins. It's called a nucleosome, and the DNA wraps around that. Okay. And it was a change in the histones that mediates this process called silencing, this epigenetic process. Okay. And that change in the histones is known to be due to a modification of the proteins. Okay. And proteins are strings of amino acids. Okay. And certain amino acids, and a good example is lysine, for example, can also be modified by a tag that can be put on or off, taken off of the lysine. And that tag is a very simple 2-carbon acetyl group that can be put on the lysine or taken off the lysine. So the lysine can be either acetylated, where the tag is on, or deacetylated, where the tag is off. Okay. Now, it turns out that when histones are acetylated, they tend to promote an open structure in the DNA. So the structure of DNA around the histones is elongated, and the genes can be expressed normally. However, when the histones are deacetylated, the lysines are taken off. The acetyls are taken off of the lysines. Okay, then that region, which is deacetylated, shuts down and forms a compact structure and renders those genes inaccessible, and they're not expressed. So that was known. Okay, so a lot of people had before us tried to see if these sirtuin proteins had the ability to remove these tags from the lysine. So it would go from an open structure in the chromatin to a closed structure, and from the active state to the silent state. Okay, so that would make it an enzyme that would deacetylate, pull off the acetyl groups from the lysine. So people tried to find this activity, deacetylation activity, in SIR2 and failed. They couldn't do it. Okay, so that's where we started, and we also didn't see it. But we were at the same time interested in whether sirtuins were silencing in a way that was somehow tuned to the metabolism of the cell. So we're also interested in molecules that were involved in metabolism at the same time. Okay, so that brought us to the really the last key point here, which is a molecule in cells that's called NAD. Okay, and NAD is an acronym for the chemical name, which is nicotinamide adenine dinucleotide. So it's 2 nucleotides that are put together, and we're interested in studying whether NAD might have anything to do with this process. And what we found, to make a long story short, is that SIR2 had no deacetylation activity by itself. But if we added to the reaction in the test tube now, if we added to the reaction NAD, okay, then it triggered the deacetylation of the protein. So that meant that it was an NAD-dependent deacetylase. And that's a very, very long-winded answer to your question of what do these proteins, these sirtuins, actually do? What is their biochemical or enzymatic activity? They are NAD-dependent deacetylases. And that has that finding has a lot of different implications.
So these sirtuins were discovered to be important for yeast lifespan in terms of how many times the cells divide. They've got something to do with regulating how wrapped up or unwrapped up DNA is and whether or not those genes can be expressed in a way that depends on this NAD molecule. When we think about the finding you described in yeast, that their lifespan in some sense is the number of times the cells divide rather than time per se, how is that would you say that's generally true? Is the same kind of thing true in human beings, that our cells kind of divide a certain number of times across a human lifespan, or what's going on there?
Well, I think that it may be true in humans, but for different reasons. Okay, so in humans, what seems to happen is, so we have different tissues, different organs in our bodies, and some have cells that divide all the time. Okay, the gut, skin, blood. Others at the other extreme have cells that divide not at all. For example, the brain. And so for at least some organs in the human body, cell division is not at play because it doesn't happen. So that cells don't wear themselves out by dividing, okay? But they still age, right? So this kind of process that happens in yeast certainly cannot apply across the board in humans, okay? So what about those organs that do have dividing cells in humans, like the gut or the skin or blood? Are they experiencing the same process? And, the answer is we don't know for sure, but the speculation would be no, because what's thought to happen, at least I would say one of the most prominent hypotheses for why these tissues like the gut also age and cells stop dividing is because of what happens at the ends of human chromosomes, where the ends of all of our chromosomes are capped by a structure that are called telomeres. And what happens in mammals, including humans, is that as cells divide, the telomeres get shorter and shorter every cell division, okay? Until eventually they become critically short, okay? And the cell senses that as essentially it looks like a break in the DNA to the cells, the end of the chromosome, because it has just this little stub now, okay? And the cell gets a signal, the DNA is in bad shape, I can't divide anymore, okay? And that's probably the most likely reason that for organs with dividing cells, cells stop dividing.
And so are those things reversible, or does it move only in one direction?
Yeah. So for yeast, we never really demonstrated reversibility. What we saw is we could make cells live longer, so we could slow this process of aging, okay? But we couldn't take an old cell and make it young again, okay? In mammals, I think people would have said the same thing until, about 10, 15 years ago, when it was shown by Yamanaka that you could take a fully differentiated cell of a human, and it could be a young person, it could be an old person, doesn't matter. And you could reprogram that cell to go back into a state of that's found in early embryos during embryonic development. Okay, and that state is kind of a stem cell-like state. In fact, they're called embryonic stem cells that occur very, very early in embryonic development. And what he did is he showed that by adding a particular cocktail of genes that were expressed into proteins into this old differentiated cell, he could convert it back to a cell like embryonic stem cells. And they're called IPS cells, for induced pluripotent stem cells. So that's reversing aging at the cellular level, okay? And the other thing we know that must be the case is as a species, and we knew this forever, we have to be able to reverse the aging process, because any species that could not do that would die out very quickly. And we continue to propagate as a species. So something about going, through the process of generating germ cells and having the germ cells make a zygote, which develops into an embryo. Something about that process as a whole has reversed aging.
Interesting. And is there, so essentially every time you create a new zygote, you're obviously creating a new organism that's young.
That's
and
young, exactly.
All the way down to the cells. They look and act young. Are there any, there's a few different places I want to go, but, that reminds me of questions related to I think everyone understands we inherit our genomes, the sequence of the genetic code from mom and dad. Can we also inherit any of these epigenetic modifications? And is there anything, so even though, when you create the new zygote, you're sort of restarting a brand new organism in the young state, are there nonetheless ways to inherit? Like if your mother and or father are older and they have perhaps accumulated mutations or epigenetic modifications in the sperm or the egg, can that type of thing lead to heritable consequences for the offspring?
Okay, so certainly genetic changes will always be heritable. They'll always be inherited. And so that, as a rule, if you have a progeny from older parents, those progeny are more susceptible to inheriting genetic changes. So from dad, spermatocytes, the precursors of sperm, replicate continuously. And so an older person has had sperm cells that have undergone more DNA replications and have had more chance for mutations to occur. And so there's a higher probability of mutations being inherited in the progeny. For mom, the oocytes don't divide during growth and development and adulthood, but they're sitting around. So things can happen and damage can occur. And we know that birth defects, many due to chromosomal abnormalities, go up with the age of the mother and the newborns. So those changes happen, but epigenetic changes, I think there's not, it would first of all be pretty hard to show that experimentally, as opposed to a DNA change where you just read off the sequence of the DNA. And I think the evidence is, at best preliminary as to whether epigenetic changes could be inherited. The other problem is, epigenetic changes are pretty much, the slate is wiped clean during formation of the germ cells. I mean, that's kind of thought to be the mechanism by which you can get back to youth again, is to do that. So that would say you would not expect epigenetic changes to be inherited generally. But I think the jury is still out on that question.
When we think about some of the key players here that you mentioned, the sirtuins and NAD and things, what is the natural course that those molecules take throughout the lifespan of the average organism? Are they decreasing or changing in some way over the lifespan? And if so, what actually drives that?
Yeah, it's a very important question. And so for the sirtuins, let me give a little bit more background. Humans have 7 of these proteins, and they're encoded by 7 different genes. They're all related to one another, but they're not identical. They're different. They all have the same enzymatic activity that I mentioned earlier. They require NAD, and they deacetylate histones and other proteins in cells. Now, the seven are different in a few different ways. The main difference is they're spread across different parts of the cell. So three of them are in the nucleus of the cell. So in the nucleus of the cell, they can deacetylate. They have substrates, if you will, or targets that include the histones, as I mentioned, which are on the DNA and in the nucleus of the cell. But it also includes proteins that regulate transcription, so-called transcription factors that turn genes on and off in a gene-specific way. And those are in the nucleus. And those can be deacetylated by the nuclear sirtuins. Okay, and 3 sirtuins of the 7 human proteins are in the nucleus: SIRT1, 6, and 7. Okay, 3 others are in a specific organelle in cells called mitochondria. And it's a very provocative thing, actually, because mitochondria make energy for the cells. And energy in cells comes in a chemical called ATP. And the ATP is produced by mitochondria. So the fact that 3 of the 7 sirtuins are in this one tiny space in the cell, I think, is at least suggesting that mitochondria and energy production might be especially important in the aging process in mammals. Okay.
Yeah, that's interesting. I was just talking with someone about mitochondria, actually, and we got into some aging stuff. So as far as the question of why sirtuins would be in mitochondria, is it because the mitochondria have their own little mini genomes? Are they doing the same kind of DNA modifications that they are in the nuclear genome?
I don't think so. I think that certainly the mitochondria do have their own tiny genomes. But from what we know about the sirtuins that are there, and I think the most prominent one is called SIRT3 in the mitochondria, is that it's deacetylating enzymes in the mitochondria, proteins in the mitochondria that either are involved in metabolic processes in the mitochondria, and the main one that seems to be a target of SIRT3 is the degradation of fatty acids, so the oxidation of fatty acids in the mitochondria, where enzymes that catalyze oxidation of fatty acids are substrates for SIRT3 to deacetylate them, and that turns them on. That makes them more active. So SIRT3 would upregulate fat destruction in the mitochondria, and that produces energy, of course, when you degrade fat. That's one way you produce ATP. Another thing that SIRT3 deacetylates in the mitochondria is an enzyme that fixes oxidative damage to proteins in the mitochondria. Okay, the enzyme is called superoxide dismutase, or SOD. And SOD is deacetylated by SIRT3 inside the mitochondria, and that increases the activity of SOD. So that would give it a mitochondria greater capacity to repair any damage that is accumulating, which may also slow down the decay of mitochondria and the aging of mitochondria. So in the mitochondria, I think there are 2 things at least going on. One is they're involved in metabolic reactions, such as the degradation of fat. And secondly, they themselves get older and more damaged as we get older. And both of those processes tend to be counteracted or regulated by SIRT3. One is to upregulate the degradation of fat, and the other is to increase the repair capacity of the mitochondria. So you might imagine then that with this NAD-dependent set of proteins, the sirtuins, you can maintain healthy mitochondria for longer if you keep those proteins as active as possible. And you might also have a beneficial effect of degrading excess fat to exchange fat for ATP.
I see. So if these sirtuins are NAD dependent, I think an interesting question is, to what extent do NAD levels change throughout the lifespan, or even throughout, like, say, the day, sleep wake cycles, diet, things like that?
Yeah, that was your original question. That I just said was background to answering your question. So, what NAD changes are extremely important in the picture here, and the most important thing about NAD levels is that they seem to go down as we get older. We lose about half of our NAD during aging, and that's going to have really disastrous consequences to the activity of these sirtuins and any other process in cells that requires NAD. So, maintaining NAD levels would be, I think, a very important way to stave off some of the effects of aging. And there's a lot of data to back that up in the laboratory. Rodents, for example, can be made to live longer if you boost their NAD levels. Okay, so that's a really important finding. And at least part of that effect is due to sirtuins, because if you deactivate SIRT1, one of the sirtuins, this effect is ameliorated. It's abolished. So I think that NAD decline is probably a very important aspect of human aging. And this was really the one of the very early underpinnings of Elysium, when the CEO, Eric Marcatulli, and I first started talking about 10 years ago about ways that a company could start to utilize some of the new findings that were coming in regarding the aging process.
And so, going to those mouse experiments where people were able to extend the lifespan of rodents, two questions there are: A, how did you actually increase NAD levels in the animals? Was it through dietary supplementation or engineering a specific kind of mouse? And B, what is the magnitude of life extension that was able to be achieved?
Yeah. So the simplest thing would be to just feed NAD to the mice. Okay. The problem with that is NAD itself is a compound that does not enter cells. It can't get into cells. So it can't raise NAD in cells if it can't get inside. So what was done is to use molecules. So the way cells make NAD is through a series of enzymatic reactions in cells, where you start with a small building block, which is nicotinamide or nicotinic acid, and enzymes make it bigger and bigger stepwise until you end up with NAD. And so what's done is you take one of the intermediates in the middle of that pathway, okay? And there were two intermediates right in the middle of the pathway. One is called NMN, and the other is called NR, okay? And the NMN stands for nicotinamide mononucleotide, the NR for nicotinamide riboside. And if you add those compounds, they are taken up by cells. So what you can do is you can feed compounds into the middle of this pathway, which will then interact with the enzymes that carry out those terminal steps in making NAD, and that will raise NAD levels in cells. So that's what was done. It was to give these compounds, either NR or NMN, to mice in the drinking water or in the food, okay, and then measure to see that NAD levels were raised, and they are, and then to see what happens. And what happens is they live longer. Now, you asked how much longer. So it turns out in mice, you can various genetic interventions can make them live longer, but only by about 10 or 20%. That's really the best anybody has done in mice. Another thing that will do that is by restricting the food in a process that's called calorie restriction. That's been known for about a century now. And again, they'll live longer by up to maybe 30% or so by that intervention. Okay. And the effect of the NAD supplementation, is of the 10 or 20% nature in extension of lifespan.
And doing lifespan studies in humans, I imagine is difficult, or at the very least expensive, and requires a lot of patience. But is there anything that's been done in humans, any studies that might suggest that something similar could be true?
Well, I don't think so, not yet. And the problem is exactly what you say, that, who's going to wait to do a human study? So what you can do is you can try to find some kind of surrogate for how long someone lives. And that's what people have been looking for a long, long time. They're called biomarkers of aging. And if you had a biomarker of aging, then you could simply look at that biomarker, and that would tell you the biological age of the organism. And you could measure that and see if you slow down its progression and not have to wait the entire lifespan to see whether there's been any effect. So that's been one of the holy grails in the aging field, is to find a biomarker for aging for a long time.
And are there any good biomarkers in humans?
What's happened, I think, at least in my opinion, is in the past decade or so, there's been a very important biomarker that's been found. And it's, again, it has to do with a modification of an important molecule in cells. In this case, it's the DNA. And it's DNA methylation. So DNA tends to be methylated at some positions where you have a C and a G nucleotide residue together. So C, phosphate, G, CpG. Okay. And you get methylation on the C residue. And that happens at various sites across the genome, maybe, I don't know, maybe 1 million total sites across the entire genome. And what's become available is the ability to very accurately assess the methylation status of all of these residues, which is a combination of chemicals that can distinguish between the methylated C and the unmethylated C, and cut the DNA at the one and not the other, and DNA sequencing, so you can tell where this has occurred. And in so doing, you can assess simultaneously the methylation of many, many residues across the genome all at once in one experiment. So that was what was done initially by Steve Horvath and his colleagues, was to just look at the methylation of DNA, okay, in various cohorts of people that were involved in human studies of different kinds, okay, so that there were biological samples available. So let's say blood of these people. And what they did was to assess methylation status across many of these sites in the genome, okay? And they asked whether they could see a pattern of methylation that changed in a very characteristic way with the age of the person providing the sample, okay? And they used, and what they did is you just train the computer to the task of finding these methyl sites that changed characteristically or monotonically with aging of the person in question. So let's say the older the individual, the more methylated that C was. Okay, so it goes from in a young person being unmethylated, in a middle-aged person partially methylated, and in an old person fully methylated, let's say. And the technology is such that you can actually make those determinations, okay? But you need the computer and machine learning to then go into that data set and actually find the relevant methyls that you could survey in this way and come up with a score that would tell you the age of the person. So that's what they did. And in the initial studies of about a decade ago, there were some 300 to 400 methyls, specific sites now in the genome. And assessing those methyls gave you a readout of the age of the person. Okay? So that's not what people were thinking about something much simpler when they were thinking about biomarkers of aging, right? But none of those simple things really worked. So here's something that looked like it worked. And, you could program the algorithm to do this task. Okay. So I think that's now been elaborated on quite a bit in the last decade. And people have really, I think, taken advantage or are starting to take advantage of this technology of DNA methylation assessment. At Elysium, we got very interested in this some number of years ago to see if we could make this work in humans, and rather than require blood, we could do it with saliva, and that's actually works, and it's a product that Elysium offers called Index.
So there's a way for scientists to take a biological sample for people and look at the DNA, but they're not looking at the sequence of DNA. They're looking at the pattern of these epigenetic modifications on the DNA. And that tends to tell you something about the biological age of the person. And my understanding is, part of the point of this is the biological age as you see it in these methylation patterns is not necessarily going to be equal to the chronological age.
That's right. It will broadly it will, right? So you'll see if you look as a function of age, you will definitely see a relationship between chronological age and this biological age. They'll be similar, okay? But they won't be exactly the same. And they'll be, imagine a straight line going age versus methylation status, okay? And each person will be a little bit off that line. Some will be right on it. They'll be average. But those that are off it are interesting because that means that their biological age is different. May not be extremely different, but is somewhat different from their chronological age. And so a test like this, you can impute a rate of aging for that person. So a person that's right on that line, okay, would be aging exactly average, okay, in the population. And you could call their rate of aging as 1, okay. Someone whose methylation status showed a biological age younger than their chronological age, might be aging at a rate of, say, 0.9. And of course, that's desirable.
If you took a random sample of a bunch of people, what percentage of people will tend to be very high off of that line, however we define that?
I think if you took a random sample, this is what they did in the original experiments, right? The way the line is drawn, right, the line will be average. So there'll be the same number of people above and below the line by the nature of the beast, okay? Now, we've noticed in customers of Elysium that there's a slight skewing so that there are more people than it would be expected that are aging more slowly
Mm.
than you would expect in a sample that size. Okay. And the way we interpret that is maybe, our customers are kind of self-selected as people who are very health conscious,
Mm-hmm.
right? And in aggregate, would show a slower average rate of aging compared to the general population.
And can you talk about how this product works a little bit more? So if someone purchases it, like what is it physically that they're doing? And then what does the output actually look like that they see in the end?
Yeah. So the product would be not terribly different from other products out there like 23andMe or Ancestry.com, where you get the product, you create a sputum sample, and you mail it off to our labs, and the sample gets analyzed, and the data gets sent back to the customer. It's extremely simple, with a lot of information to put things into context for the customer. And so I think it's a cool product. I think it's really cool though that it now potentiates, it allows you to do other things. So for example, our first product, which was based on the research I talked about earlier with sirtuins and NAD, is called Basis. Okay. And it has two ingredients in it. One is an NAD booster, this thing NR. It's one of the ingredients. And the second ingredient is based on research that shows that there are compounds that specifically activate one of these human sirtuins called SIRT1, which is a quite important one, actually. It's a nuclear sirtuin. And the original studies showed that one of these activators of SIRT1 was a molecule maybe some of your listeners would have heard of called resveratrol.
The red wine molecule.
The red wine molecule, yeah. And there's a lot of studies have been done on resveratrol since David Sinclair originally showed it could activate SIRT1. And in rodents, resveratrol has a really good effects on the health of the animal. It really is a amazing molecule. And the thinking 15 years ago was that this was going to be wonderful for humans. And a lot of people, including myself, were taking it at that time. But it turns out in humans, its so-called bioavailability is not so good. So it's very hard to get an adequate dose of resveratrol in humans. And so we knew all that 10 years ago. So we opted for a more obscure compound that comes from blueberries, not grapes, which is a structural analog of resveratrol. Okay, so it's similar as a molecule, but with some differences. And we had noted in the literature that even in animals in the laboratory, it seemed to work at a lower dose than resveratrol, which says it's more potent. And so we think that this is a version of resveratrol that is more bioavailable and much more likely to have beneficial effects in people. And it's called pterostilbene with a PT at the beginning. And so that's the second ingredient in Basis, this combination of NAD boosting, which should raise the activity of all 7 sirtuins, along with pterostilbene, which should, by a different mechanism, activate SIRT1. We thought that was probably from what we knew at the time, and would still be true, will give you the best shot of maximizing the activity of these sirtuins, especially against forces of nature that are causing NAD levels to decline and deactivate them as we get older. Okay, so that's the product. That's the rationale behind the product, that it contains two kind of synergistic compounds that give you the best shot of activating sirtuins and impacting aging favorably. Okay, so now you want to, what we're interested in doing, and I just give you a little teaser about this, is to ask, can we use these two products of index, DNA methylation product, and basis, the NAD sirtuin product, can they play off one another? Okay. And so one thing we've done is we've looked at, we had samples of people from one of the studies that we've done, actually, where we could take those samples and do both the index test on the people and also measure NAD levels in these people. Okay, so these people have not taken basis; it's just people out there in the population, and we found that there was this relationship in that the higher. So we all have different NAD levels; they're not going to be identical, right? It's determined by who we are. And the higher the NAD levels, what we found, the pattern was the higher the NAD levels in these people, the lower their biological age. That's determined by index, which is really good, I think. And so it I think, establishes credence for the idea that there really is a relationship between NAD and aging in humans. Okay, so the next step, which is a study we have started to do, is to ask, okay, so what happens to somebody if we measure their biological age and they start taking bases? What happens to it? I mean, so this gets back to something you asked much earlier about reversing aging. Okay, can you reverse it? And that's a study that's going to take time, because now that's the so-called longitudinal study, where you're following the same people over a prolonged period of time. So we don't know the answer to this yet. And my suspicion is, we started this several years ago, like 3 years ago. And, we started looking at the data, and my thinking is we got a confound that occurred in the time frame of that study. And for this, we just simply ask our customers if they're interested in opting in to something like this, where we simply, at baseline, before they start, new customers, before they start taking basis, they do an index test. And in a year after taking it, they do another index test. Okay, it's that simple. But what intervened in that year was COVID and isolation protocol and change in lifestyle and habits. And we suspect that was a major confound, that it will probably be shown that COVID changed the rate of aging in people. I really expect that to be found. So we very much are interested in getting good data on this question, but we don't have it yet.
Interesting. Given everything everything that we know about aging so far, do you think it's possible, in principle, that one day we could basically put an end to aging to be effectively immortal, such that the normal aging process doesn't happen, and the only way people would die would be if there was some other kind of disease or accident or something like that?
I do.
Oh, wow.
I do. And, I would have said no up until as recently as 5 years ago. And I did say no anytime I was asked that question. But I've reevaluated. And I think that at some point, if civilization continues to advance in the way that it has been, and nothing catastrophic happens to the world, that it will be possible. Part of the reason for saying that is that, you can achieve these little bits of success with things like NAD intervention. Okay. And, many important discoveries start with something that sort of barely works, and then you make it better and better and better. Okay. And the other thing relates back to another topic we discussed, and that is that it seems you can reverse aging at the level of individual cells by converting skin cells, let's say, of an adult animal back into embryonic stem cells. Okay. And if you can do that, make that complete reversal, right? Why shouldn't it be possible to make a little bit of a reversal, at least enough to stop aging in its tracks? Okay. And there's been some papers published in mice that would lead you to believe that this might be possible in humans. And it's a little bit, you can do things in mice that you can't easily do in humans, of course. So this process of turning cells back into stem cells that was developed by Yamanaka involves putting into those cells 4 different genes. And those 4 genes, when they're expressed, will turn the cells into embryonic stem cells. Okay. And so in mice, of course, you can make transgenic mice where you've added genes into the genomes of the animals. And so what was done is to put those 4 genes into mice in a way that you could turn them all on at the same time in all the cells of the mouse. Okay. And initially, when they did that, in the first set of experiments, what do you think happened?
Tell us.
They got cancer.
I see.
And the reason they got cancer is these embryonic stem cells. Remember, this is the early embryo, which has to grow a lot. And these embryonic stem cells are very rapidly growing cells.
I see.
And if you culture embryonic stem cells, they grow like gangbusters. Okay. It's like you're growing a bacterial cell that grows so fast. And so to just do this in an unregulated way won't do in fact, it'll do the opposite of what you want to have happen. So what they then did was to figure out a way to control their expression, these 4 genes, so that you can give the animal short pulses of intermittent expression. So you give them a pulse, wait a few weeks, another pulse, wait a few weeks. And when they did that, they reported elements of reversal of aging in the animals, which is a very striking finding. So, there's just been a few papers at this point in time on this. So I think we want to wait and see and assess this more completely. But it at least raises this possibility that this is a strategy that could work. Now, in humans, of course, you can't simply, start mucking around with the genome in this way. So you'd have to find some other way to skin the cat. But, those are technical problems and not problems in principle.
Well, another thing that this makes me think of is, questions related to the brain, which I wanted to talk about. So, even if you could reverse aging through some of the means that you've been describing for us, if you were to, your neurons are really weird in certain ways that we touched on a little bit earlier. They don't divide. And yet our brains do age. And not only do you want to preserve longevity and have youthfulness in terms of the functioning of your mind, but you also couldn't, even if you wouldn't want to, even if you could, to sort of revert all of your neurons back to a stem cell state to rejuvenate them, because you would lose all the information that makes you you, and that is your life. So how do you start to think about aging in the brain, and how, is there anything known about how to preserve the function of neurons or prevent it from decaying as quickly as they normally do?
Yeah, it's a difficult challenge. So the first question is, what actually happens in the aging process of the brain? And years ago, it was thought that we simply lost neurons, that neurons start dying and the number of brain cells goes down. And that's how the brain ages. And consistent with that, people found that the volume of the brain shrinks with aging. And so that fit this neat idea that you just lose brain cells. And since they're non-dividing, once you lose a cell, you cannot replace it. Okay. Now, however, that's probably not exactly correct. Okay. And probably more important than the loss of neurons. So neurons are lost, but in particular in neurodegenerative diseases. And the loss of neurons really, it tends to, there's a lot of overlap here between normal aging in the brain and disease-induced aging. But in Alzheimer's, there's neuronal loss. But in normal aging, there probably isn't. And it's probably more a matter of just shrinkage at every level in the brain, at the cellular level, at some ultrastructural level, but not at the level of the neurons actually die and disappear. So you have to preserve more than just the neurons in the brain. You have to preserve the connectivity of the neurons, which are connected at synapses that are due to processes that extend from the neuron, which is where really the information lies in the brain. And so it's a very difficult problem to think about. We can begin to address some of this. So again, we've been interested, I'm interested in the brain myself, and Elysium has been interested in this for a while. And so one study that I think is a very good study out of Oxford University studied aging in the human brain in subjects, and it was monitoring aging by brain volume, which was determined by imaging. And so the study really leverages advances in brain imaging technologies, which has gotten quite good and quite quantitative. And so it was a 2-year study, and they could clearly demonstrate significant shrinkage in the brains in this poised population that they were looking at. The poised population was, I believe, people in their 70s who had mild cognitive impairment, so MCI. So they were, at a point where they were losing brain function, perhaps a little bit faster than ordinary people, which made the study doable. And so they could see shrinkage in the brain. Now, there was some information out there that there was a bad actor that's produced that's causing at least some of this shrinkage, which is called homocysteine. Okay, and what you could see is that there's a relationship between levels of homocysteine in people and brain shrinkage. And so it was thought that if you could reduce homocysteine levels in people, you could also mitigate the brain shrinkage. And it turns out that they showed that the right combination of 3 B vitamins, of all things, at the right doses, which tends to be much higher than the doses you would take in a vitamin pill, could reduce shrinkage in people, brain shrinkage, over this 2-year period. And part of the reason is that I don't remember the exact details of this, but one of the B vitamins, I believe it's B6, B9, and B12, one of them promotes the degradation of homocysteine, and the other two retard the synthesis of homocysteine. So both have an effect together of lowering homocysteine levels in these people. And so that's an interesting finding. And, we got involved with David Smith, who conducted this study a while ago, and have based another product at Elysium called Matter on, the exact formula of what they were providing these people. Now, the only thing the only limitation of the study in the people is it showed that people who also had unusually low levels of omega-3 fatty acids didn't respond well to the B vitamins. They didn't have an effect. And so it's thought that, in order to get the benefit of this, you need sufficient levels of omega-3s. So we put an omega-3s into the product. So everybody taking the B vitamins will also be getting a dose of omega-3. So that's, I think that's an interesting product because it's based on human studies and can impact the brain in a way that we think is meaningful. That is to say, the volume of the brain.
So what are all, remind us, what were all the ingredients in that product?
So it has the 3 B vitamins, B6, B9, which is folic acid, and B12 at the appropriate doses.
And what are those doses compared to
they're higher. They're much higher. I don't remember the details, but they're much higher.
I see.
And for those people who might not benefit by virtue of having low omega-3 fatty acids, which is in itself not a good thing. Omega-3s are healthful. There's an omega-3 supplement in there.
I see. So that one, that's called Matter. That's like the brain product. You've got one called Basis, which is basically the NAD product that we mentioned earlier. And you've got a couple other ones. What are those, and what's the thinking behind the ingredient list there?
Yeah, one of the newer ones is called Signal. Okay, and that's related to Basis. Okay, so the NAD booster in Basis is something called NR, nicotinamide riboside, and that's essentially a half of an NAD molecule. So you have the nicotinamide nucleotide and a ribose. That's NR. NMN is one step further down the chain, and it's the nicotinamide and the ribose with a phosphate on it. So it's one step closer to NAD. Some people favor nicotinamide over nicotinamide riboside, although in animal studies, they both seem to work equivalently. Okay. But there was a paper published on NMN fairly recently showing that it had the ability in humans of making insulin work more efficiently. That is to say, increasing insulin sensitivity, which is thought to be a good thing for health. So we wanted to have a product that had NMN in it. And then we found, so one of the other interesting targets that, I've been interested in for a long time. So pterostilbene targets SIRT1, as I said earlier, as does resveratrol. But there's this important sirtuin in mitochondria, SIRT3, that we think will be important in maintaining mitochondrial integrity and health with aging, if we could keep it active. Okay. So the NAD booster will help with that. But if we had a more specific molecule that could target it, that would help even more. And so the literature informed us of a molecule called hinokiol, which is reported in several different labs to activate SIRT3. And so again, that's something we got very interested in and obtained a very good source of it. And that's the second ingredient in Signal. So we think Signal will have an effect that will be more targeted on mitochondria in aging and energy production, things of that sort, perhaps fat degradation in mitochondria. So that's, again, on the metabolic side with basis.
So you mentioned earlier, I think an important thing to talk about is bioavailability. You mentioned, for example, resveratrol is a very interesting molecule that does really cool stuff, but it's not particularly bioavailable. And that was why you guys were using this other thing called pterostilbene, which is more potent. Are the ingredients in all of your products reasonably bioavailable, or do you add something to it to boost the bioavailability? Because I know that my understanding is a lot of supplements out there, not in every case, but in some cases, it might say it's got 500% your daily value of whatever, but you end up absorbing very little of it. So how do you think about bioavailability?
Yeah, we're interested in bioavailability in all cases. And, so I think that in the products we've discussed, the compounds are relatively bioavailable. Where they're not, then we're really interested in technology which exists in which the compound is essentially encapsulated in vesicles to make it more bioavailable. And I think, the vesicle technology, again, is one of the things that I think has really taken off. In fact, the RNA vaccines that deliver RNA to cells work because the RNA is first encapsulated in vesicles. And the actual vaccine is the vesicles that will be stable, in the blood, and will then be able to fuse with other the membranes of cells to deliver their contents to the cells. And so we're applicable, and a number of the products we're thinking of that are in development, we're thinking of starting to use this technology to increase bioavailability. So yes, I do think it's an important issue in any of these products. And I think it's one that will become increasingly utilized by us down the line.
And I am just curious too. So you co-founded this company called Elysium, which is making these products. On the one hand, I can imagine one thing that prompted you to do this was just your research and the relevance of what you were learning about aging to making these kinds of products. But what made you want to start a company as a scientist? I think most scientists I've met aren't inclined to do that.
Well, first of all, I think it's MIT is supportive of their faculty also being entrepreneurs. So that there's a supportive environment that we have. And a lot of my colleagues at MIT start companies. And in fact, I've been involved in companies in the past. Most recently, before Elysium, I was involved in a sirtuins company called Sirtris with David Sinclair and Christoph Westphal. I was co-chair of their SAB. And since the start of Elysium, I've been a founder in another company that's a kind of a drug development company to target SIRT6, another important sirtuin in the nucleus, but is really highly focused on clinical drugs and developing drugs on SIRT6. So I think that it's basically for aging. There was all this work that had been carried out over a long period of time that was just beginning to make a blueprint for how you could take this and develop it into something that could benefit health. And so it's a very attractive prospect to try to do that and to see can we first of all find out will this work the same way in people, and secondly have the satisfaction of actually being able to impact human health with such a company. The other thing I liked about the concept was really Eric Marcatuli's idea that, on the one hand, at the time we initiated our conversations about 10 years ago, there were these supplement companies, of which there were many, The quality was really, frankly, not great. And some of them didn't even have in the product what they claimed was in there. And if it was in there, it was almost certainly something that didn't do anything. And so we wanted to make sure that we had products of high quality, and thought that if they really worked, if we could assure and do the testing in humans to back up the preclinical data that they really worked, that it would actually start to form a space between the supplement companies on the low end and the drug companies here, the classical drug companies, that there was a space in the middle that was an opportunity space to have compounds that really worked, but that could be delivered to customers quickly because they were natural. And so they didn't have to go through the kind of FDA testing and approval of a novel compound, a drug. And that was an exciting possibility to have this platform, be a platform company that was not a supplement company, that would be able to essentially have a rapid timetable for development and testing and delivery of products direct to consumer online in an efficient way. So, there's kind of a lot of attractive features about the company for me. Its novelty, its prospect for saying that, this aging research really led to something, and targeting aging is really a good thing for health.
I know the aging field is really booming right now. What do you think what are one or two like big outstanding questions in aging right now that you think we'll likely know the answer to in the next few years?
Well, I think something that's really, there's a lot of studies now on senescent cells, this area called cell senescence. And cell senescence is when cells stop functioning, doing their normal function, but they don't die. So they hang around. And that's thought to be a bad thing. And it's thought that if you could take those cells and kill them, eliminate them, it's much better than having them hanging around. And so there are companies out there, a lot of them, which is why I think we're going to know about this quickly, that are developing compounds that can selectively kill a senescent cell. Typically, these senescent cells, so I mentioned earlier that when dividing cells divide and their telomeres get sufficiently short, right? They get a signal, a damage signal, okay? And they stop dividing. They can also get that damage signal, not just by telomere shortening, but if the DNA actually does get damaged as the cells get old, and they stop dividing, but they don't die. They just sit there. And so these companies are developing these senolytic compounds, so it's compounds that can lyse senescent cells. And there are, a lot of tremendous amount of activity in that field now, which is why I think in the next few years, in a very short time frame, we'll know the full potential of this area of aging research, that you can impact aging favorably by targeting not an old organism, but old cells in the organism.
Interesting.
Yeah.
Well, Leonard, are there any final thoughts you want to leave people with, or anything you want to emphasize from our conversation?
Well, I think I would like to leave people with the idea that we've learned a lot about aging in the past 3 decades. And typically in research, the benefits of the research lag from the time of the research itself. And we're seeing this in cancer, where incredible information emerged about human cancers from the 1970s through the 2000s, but very little in the way of treatment, with the exception of childhood cancers, and in some cases diagnostics for cancer. But medicines lacked. for cancers. But I think that that's starting to change now because a lot of work's been done to take advantage of the basic science. And the same is going to be true in aging research, okay? But it's staged, about 30 years later than cancer research. So I think people should take heart that there has been progress and that there will be remedies, not to eliminate aging, but to impact it in a way that we can stay healthy longer and keep doing what we like doing for a longer period of time. So I think that's one take home I would like people to have. Another is that, I think that there's kind of a seamless continuum of the academic research environment and the world of companies where, you want to go, if you can be on a path that starts with a simple experiment in the laboratory and follow that all the way to human studies, it's a very satisfying thing. This is a personal statement, and it's been great to be a part of that all along the way.
Well, Dr. Leonard Guarente, thank you for your time and everything that you shared with us.
Thank you.